So today we are going to do a cell disruption method by glass beads. So for that you need to take 500 microliter of the sample for 6 tubes. So this is a glass beads which we are going to use. So approximately we are going to add some 350 microliter of this. So for that we are going to take 5 beads for each tube. So we have taken in spatula. So equal number is very important for equal volume of the beads. So we have taken now beads in all things. So now the next step we are going to press this. So once you have added the beads, now you need to mix that. For that we kept the samples in Vortex machine. You can see 5 samples, each 2 minutes interval. So the one which we are having blank, which is not have added beads, as well as we are not going to keep it for the mixing. So now you can switch on that in a constant mode, just to hold your hands in the top, so that the tubes are not popped out from that. So like that you need to do for individual timing up to 10 minutes. After the 10 minutes is done, so you have taken all the samples. Now you subject to do centrifugation. So that at the end of the centrifugation we are just to collect the supernatant. For 10,000 rpm. RPM. Yeah. So we have kept samples 10,000 rpm for 10 minutes. So we have to wait. So after centrifugation, we have collected the supernatant. Now you need to take a OD at 280 nanometer for protein estimation. So for that, first you need to keep the blank. So now you have set for the blank. So after the blank, now so we are going to add the sample 1, that is 2 minutes. So like that you need to take the reading for the remaining samples. So like that we have to measure the OD for all the samples. So here the readings. So starting from sample 1, that is 2 minutes, then 4 minutes, 6 minutes, 8 minutes, and sample 5 is 10 minutes. But the last sample is the 0th minute, that is not subject to any mixing with beads. So here are the samples. So now you need to proceed like your experiment number 1 and 2. So you need to calculate the protein concentration for the respective ODs. And you need to draw the graph between concentration of protein in y-axis and the time of mixing in your x-axis and find out optimum time.